Welcome everyone to this SymbNET series. My name is Massimo Micone. I'm a PhD student in the Evolutionary Biology Lab here at IGC. We have two speakers. The first one is Jan van der Meer. I hope I pronounce it okay, from the University of Lausanne. And the second is Tina Keller-Costa from the Instituto Superior Técnico in Lisbon. Yeah, perhaps I can start. So Jan is a currently full professor in environmental microbiology at the University of Lausanne, where he's also head of the environmental and evolutionary microbiology group. His academic path and interest developed around the study of environments and their relationship with their microbes from, I would say, a fundamental perspective, but also with the idea of trying to use microbes to improve the quality of the ecosystems we are also part of. And today he will tell us more about the soil microbiomes. The stage is yours. Welcome again.
Okay, thank you very much. Is everything visible and can you hear me all right?
Yes.
Good, thank you very much. So it's a real pleasure to talk to you today. I know you're a consortium obviously from Philipp Engel, who is member of it, and who's also a member of a bigger research initiative in Switzerland on microbiomes. That's called the NCCR Microbiomes, which I'm currently directing. So my own interests lay in soil microbiomes, and particularly also in polluted environments. And this has been since a number of years, this has been a research activity because of the fact that we know very well that polluted soils, they degrade very quickly in their capacity to sustain plant growth. And so there is increasing interest to actually try and manage this or restore this to some level, but we have all understood also that this is extremely complicated. Luckily, so there's more and more interest in this kind of management and restoration of soil microbiomes, and I will tell you today three parts of that. Before I continue, I would like to introduce you the people in my lab who are currently working on it. This includes PhD student, postdocs, and senior scientists, but I will be particularly talking about some of the aspects from Marianne, who was a PhD student, and Manu Priyam, who was a PhD student, and Senka Čaučević and Tanya Miguel as well. So let me introduce you soils. I'm sure that you're familiar with soils because normally we walk on it. I'm sure that Portugal also has quite a few different soil types and so on. It's important to understand that soils, in terms of being a microbial community, is a living environment. It is considered the microbial community that is the most species rich. If you think about soils, and it's not really clear whether you could say, okay, this is a single microbiome, or if it's a single type of microbiota, or if it's just a constellation of different small pockets that are on their own, disconnected and connected. We'll get a little bit to that. What's important to realize is obviously that soils are absolutely crucial for plant growth and plant health, and therefore for the sustainable life on our planet. It's also clear that soils are being degraded faster than we can restore them for various reasons that involve climate change as well as malfunctioning agriculture practice. They're also key for a lot of diversity of animal life that live in there, and they're considered to be key for ecosystem services that go anywhere from element cycling, carbon storage, obviously, water purification, water storage, and so on. So one of the big problems that we have currently is that we realize how important soils are, and also how important microbial life is for sustaining this complex system that is soil, but exactly how soil communities form and how they maintain their equilibria, what happens if you degrade it, how we can restore it, that's not known very well. And just recently there was a new review pointing out to this in Science, saying that we really need to focus on soil microbiota, because with that we can also work on the restoration of degraded lands. And there is an awful lot of degraded lands, unfortunately, on our planet that would benefit from being restored, because then we can regrow plants and forests and so on. So this is really an important topic, I feel, as well. And I'm happy to see that also bigger journals take that up, obviously. So to introduce you a little bit to soils, there's something that is very weird, and that is shown here in the right, where at some point we sampled 100-something sites here in the Swiss Alps. And wherever we looked in these topsoils, the phyla composition was sort of the same. You can say, okay, there's the same 8, 10, 12 phyla that you find there. There's slight fluctuations, and it is known, for example, that pH plays a role in sort of determining which phyla are more abundant than others. But if you look at it from a broad thing, then you would say it's the same basically everywhere. It's also important to realize that soil is absolutely totally species rich, and with most sequencing approaches like here that is shown here on the left, where you have a typical sequencing effort to sequence the amplicons from the 16S RNA gene, you'll find anything between a few thousand to 10,000 different variants in your detection limit of the parts that you have been sequencing. Other studies have reported anywhere between 10,000, 50,000, maybe 100,000 species. And then we're only talking about detecting OTUs or ASVs, and you're not detecting any, substrain variation and so on. So this diversity is mind-boggling, and it's very difficult to understand how that actually is created. And what's also important is this small graph here that shows the cumulative proportion of species that take up the biggest part of the soil. And you can see that soil, in contrast to, for example, lake water or activated sludge, to have 80% of all the species that you detect, you need about 200 or 300 OTUs at least, whereas for other systems, this is much easier saturated. That means that most other systems are characterized by a long tail of very rare species, but in soil, you can see that the distribution is much more global. So there's much more species actually there that have abundances that are appreciable, and maybe also in that sense, play an important. Now, what do we know about the soil as an ecosystem? So your SymbNET is on host-microbe symbiosis. Soil is not necessarily a host, it's an environment. It's an ecosystem that is at the same time hosts for plant growth, for animal life, as shown here. And it's characterized by a large heterogeneity in particle sizes, in the chemistry of the surfaces, in its water content that is there. And as you progressively go down to smaller scales, you can see that so small pores, they can be filled with small roots that provide local carbon to pockets that are then filled with microbial life. And eventually, if you go down to the smallest scale of a few microns, you can have very heterogeneous and very disconnected patches of where microbial life can be sustaining. And that has obviously consequences, because as the soil is mostly disconnected, and unless it is fully saturated with water, and you can have preferential streams and so on, that means that microbial life is confined to small pockets where there may be periods where they get nutrients or not, periods where they have to sustain on parts of organic material in the soil that are very, very hard to degrade. And also, importantly, these small pockets, even though the diversity of the soil, the species diversity of the soil as a whole is immense, that means on a micro scale, this diversity is not immense, and most of the species will actually encounter maybe a few other species in those small environments. And that has some consequences, because there's now a lot of talking about, what means it's cell-cell interactions in terms of how communities are following up. So we'll get a little bit to that. Then the other thing that is very important to consider, and we have started to do some kind of a conceptual thinking about this as well with people in this NCCR microbiome. So how do microbiomes actually form? It's important to realize that soils in most cases, you don't have pristine soils that can be recolonized. In some cases, this occurs, like retreating glaciers or so, but in most cases, the soil is occupied. So you have sort of a turnover, but imagine that the soil is a de novo colonized, then probably what you have is you need an inflow of microbes that come into your system, that find some pockets, find food and substrates to live on. You can call those first arrivers. And as they start to grow, they will modify their niches that lead to subsequent formation of living habitats that then can be occupied by later arrivers. So you get like a succession during that growth in your soil. You can also have constant inflow and outflow. And then at some point when the soil is in equilibrium, which is in most cases the situation, you can still have new microbes coming in. And some of these may be invaders and some of these may not. and that has a particular consequence of their survival as well. And I will talk about this part because there is an interest actually in this kind of microbiome engineering to understand in what way can we actually bring in new bacterial species into such a system, which are then considered invaders that come into a steady state ecosystem that is full of microbes in various pockets as well. So there is this idea that, okay, yes, you can have a succession of different states, or there is a dynamic a succession of different species that live there. There's niche modification. You create a diversity of niches. There is maybe in a steady state soil community, there's very little new carbon input, except that it comes from, for example, plant roots or decaying animals or burrowing animals or so. But on the other hand, you can have extreme variations in substrate unavailability, and not that leads to, you know, cell death, cell turnover, predation, etc., etc. Now, let me tell you about 3 projects that we are currently interested in. That is, can we sort of mimic this first phase of growth of a soil? So can we do as if we have pristine soils and understand if soil microbiomes can be grown, and what happens if you try to grow soil microbiomes? Is this something that occurs reproducibly? Is there something particular in this formation or not? Then can we understand what are the major driving forces for this kind of microbiota development of complex community? And can we understand what is the role of these interspecific interactions in this, while knowing that the soil is maybe an assemblage of micro compartments or so? And how does that influence the development and maintenance of soil microbiomes? And this then relates to a third part that I want to briefly dwell on, which is, if we know this, can we understand sufficiently well to then say, okay, for example, if we have damaged soils, can we have microbial strategies to complement or restore such microbiomes with a reasonable idea of success? So I will tell you 3 small stories about this, going from this microbiome formation to then measuring interactions, and finally some of our efforts to actually try to complement and restore damaged soil systems. So we try to design a system to actually culture soil microbiomes as complex cultures by using 2 approaches. One is working from cultured soil isolates that we could get on different media and then compose them back together. This is like what we call a synthetic community, or starting from a complex mixture of cells that we haven't characterized any further a priori, that you can wash from soils, you can enrich it on things like nicrodens gradients, and then re-inoculate it into soils that we call NATCOMs. And then what we did was trying to design a system that would be similar as liquid culturing that is typical in microbiological labs. So you have a soil that is sterilized and it has nutrients in it that we inoculate with a low quantity of cells. We incubate it, so the community as a whole will grow. Then we can take 1/10 and refresh it and dilute it into a new flask with fresh soil, etc. So you can continue these transfers and see if these communities can be maintained and what's the diversity of these communities. And then we also kept soils just for very long periods of time and sampled them after 2 months and 6 months to see if that has an influence on the community trajectories. We did this in a number of different replicates to try and see, is this something that is prone to stochastic variations or not. So this has recently been published by Senka. What we see essentially is that, yes, this approach is not bad at all, although, you know, we were hoping a bit more in the sense that here you see, for example, the diversity of the initial natural community as we isolate it from the soil, and then after growing in these soil microcosms, you'll see you capture something like 30 and then in the end 60 different OTUs. There's many that go undetected, obviously, because of the sequencing approaches. And then in the longer incubated periods, so 2 months and 6 months, you see even higher diversity coming up. And more importantly for us, so we see that most of the phyla are actually represented, except a very obscure phyla that has no cultured representatives, which is called the Patesi bacteria. And nobody really knows what these are. So this told us, hey, this is an interesting method. You can, with these transfers, also maintain a certain stability of this community over and over again, which could be an interesting platform then to actually study effects on, for example, inoculation or perturbations on these communities because they are so stable. What you see here on the right low is actually a comparison to a liquid control that we did to try and understand, okay, is it the composition of these bacteria that determines their trajectory, or is it also the conditions, or is it both? And we see here that, so if you have liquid conditions, the community as a whole drifts to a different state, which is also in itself quite reproducible. And in the soil, you get another particular state that is actually driven by the community under these conditions that remains pretty stable over time and slowly drifts back to an inoculum state in the longer run. So we thought this is a very hopeful start. And moreover, we also saw that the individual replicates were quite reproducible as well. This was even more so for the syncoms, where we had 10 replicates, and there was no particular replicate signature that maintained. So they all looked alike. And you can see that over time, so here in these trends, so for example, you keep sort of the same composition over and over, and then only at the later moments you get a drift to have a more diverse composition of this community. And in the natural communities, we could see that in some cases we pick up a signature of a particular replicate here that then maintains over time and doesn't intermingle with other replicate microcosms that were carried on at the same time. So the more complex your community is from the start, the more chance you have for a stochastic effect in the beginning, and that is then propagated and maintains its own replicate signature. So that we thought it was very hopeful. And then we said, okay, how can we understand what is really happening in those soils in terms of, does it matter which species are there and how they interact with each other? And for that, we tried to do measurements on interspecific interactions, so interactions between species, a system to create random species combinations, so cell couples, that we could test then for growth effects. And typically growth effects, as I will show you, are interpreted in terms of their interactions. But here, in contrast to many other studies that have used cultivar species, here we didn't want to start with cultured species, but we wanted to start with the extracted mixture of the cells that you find from a soil and then encapsulate them in small beads that have a diameter of 40 to 70 microns. By chance, you would get two cells in there. They, because of their close distance, would influence each other or not. We can incubate that over time with the nutrients from the soil, and then we can estimate from microscopy images like how big have these microcolonies grown, and then infer whether they have interacted or not. Now, this is a bit tricky, because typically what you would assume is something that is shown here on the left. So you start with two cells over time, or maybe one, and you hope that they will grow into microcolonies. And then you want to say, like, look, here's a big colony and a small microcolony. Does that mean that the bigger one was influencing the smaller one, or the smaller one the bigger one? And typically people would refer to a null model where you say, well, you have a monoculture growth, monoculture of the other species. If we put them in mixture, you see we have 50-50% of both species in their mixture. So the null model says they have no interaction on each other. The measurements that we can do on these beads is measuring colony sizes. So for example, we can measure over time what is the one partner and what is the size of another partner. So you get this kind of diagram. But then comes the question, so can we really say that if we have one partner that is big and the other partner is small, or if they are both small or both big, whether this is neutral interactions or positive of one or negative on the other? Or not. And the problem is that we don't know what the growth rates are of these individual species. And the growth rates are very determining for this outcome. So in fact, this null model is not at all correct. And that you can see here on a simple experiment that we did with two pseudomonads. So they're almost growing to the same size, same density on the same substrate. But if you mix them, then you get one that is over proportional compared to the other. But in fact, this doesn't mean that there is an interaction. It's just that one is growing slower than the other, as is shown here in the model. Okay, so it's really important to, from this type of analysis, if you do an inference, that you know a bit what growth rates are. Unfortunately, in results, we don't know what growth rates are, right? So what we saw, for example, when we random purged these soil cells, we saw that, interestingly, all the beads, so on average, the beads that have two partners in there grow better, more than twice as good as that of singles. Okay, that is shown here in these diagrams where you have histograms that show the distribution of the microcolonies in the beads, either for beads where you have one colony, so one species alone, or where you have two or more colonies in there. And what you can compare is, for example, here in this red part, where you see that this distribution for the paired species is shifted to the right. That means they tend to be bigger than the ones that have only one and more than twice as big. So this is very significant for different experiments, different carbon substrates. Here, the soil extract, and here a mixed carbon solution of 16 different carbon substrates. And this is interesting because there was recently a new review that suggested, or a perspective, that bacterial species are mostly negative on each other. They rarely work together. However, when we look at the soils, we see that no, that's not the case. Most cells actually need a partner, and they profit from this partner in distance of each other, because otherwise they don't grow at all. So that's, I think maybe this conclusion that came out of this work was biased by the fact that they looked mostly at culturable species. So in order to try and understand this a bit better, we tried to make a model where we'd actually simulate. So we said we start with 300 OTUs, different species that are encapsulated in those beads. We know the distribution of the different species among that. So we can make a model where we have 300 species and we model 200,000 beads, for example, in which 60% are single cell starting and 40% are double cells. That is what we see at start. And then what we try to model is a different kind of thing. So we said, we don't know what the growth rates are, but we can model, are they random or not? And then compare with our experimental observations. And we found, no, they're not random. They must be determined that the ones that you find are very abundant in your sample tend to have faster growth rate than the ones that are very poorly abundant. Is there a penalty when you are alone? Yes, there is a penalty when you are alone. So when you are alone in a bead, you have on average less tendency to grow than when you are paired. Are there dead cells at start? Also, that was likely. So if we extract a solution from the soil, there's an awful lot of dead cells or cells that do not grow. So when we take all this into consideration and then look at, can we impose interaction types? And that's the advantage of a model, because I can have, you know, my 200,000 beads, I can say you grow at this rate, and now there is a factor of 0.1 that influences your growth rate, right? So I can impose different types of interactions. Is there no interactions? Unlikely. We don't see the patterns if there's no interactions. Are they random? No, also unlikely. Are there like some bacteria that tend to influence more than others? Also unlikely. Are they perhaps mostly positive on each other? Also unlikely. Are the interactions maybe mostly negative? Also unlikely. The best outcome to explain the observations is a biased model where we have slightly more positive interactions than negative interactions. And then, for example, we can compare the observations of the paired microcolonies in beads that we have on different time points, different incubations, repetitions of the experiments, with sort of this type of simulation that has these biased kind of interactions. So it really seems as if, yes, being partnered for soil bacteria is really important. On average, there's a slight positive effect, let's say 60% versus 40% negative. And I think that is important because this kind of method you can use also to characterize other microbiomes that may be different. Maybe there you find more negative interactions than more positive interactions. Okay, last point very quickly. What can we do to actually influence such microbiomes? So is there something that we can do to potentially restore communities after, let's say, a pollutant stress? We have been focusing on pollutant stress because this is something that is very important, because a lot of soils have been polluted and they suffer from it because these compounds are toxic. They come from pesticides, they can come from oil spills, they can come from other spills. It doesn't matter. There's an awful lot of danger and damage that has been done. There has been this idea that if we were able to find specific bacteria that have a potential then to detoxify a pesticide or grow on an oil component, maybe we can add it to the soil, it would get rid of that pollutant, and then the others would thrive again. So we have been looking at this for a while with different types of inoculants that we had isolated or that others had isolated. We have been looking at the question like, okay, what happens actually if you inoculate a strain into something like a polluted soil? What is different in those strains compared to, let's say, a liquid culture? We have been looking at, are there anything like specific genes that these organisms need to survive in soil or grow in soil by using transposon mutagenesis? We also have been starting to look in how these inoculants by their activity would actually become integrated into the community interaction network. Like, would there be favorable interacting inoculants or unfavorable inoculants, etc. So I cannot show you all that we have been doing, and just a few snapshots from this. Yes, we have found a number of bacterium as different as sphingomonads or pseudomonads that can actually grow in a polluted soil or a soil that you pollute, for as long as you provide that particular unique carbon substrate. So they need to have a specific niche that is shown here, for example. If you don't have a niche, then the organism grows very poorly. If they find a niche with their unique carbon substrate, it's sort of evident, huh? But still, if it finds its unique carbon substrate, then it will obviously grow. That's the same for this organism. So if we add its unique substrate, then it will grow. If it doesn't, then it doesn't grow. And that holds for a variety of soils, not always. So there's some soils where maybe there is an awful lot of predation or so where we cannot see this, but yes, they can survive and they can grow. That's important because then we can actually do experiments to try and understand what do they see. So in this particular Pseudomonas, interestingly, so we can do transcriptomics in soil. We try to compare the signals in different soils versus not completely liquid culture, but to an artificial glass matrix, so that we could see what is the heterogeneity or the particle effects and so on. And we see actually very few common changes in soil-induced transcripts. So that means that there is not, as far as we can say, there's not a particular soil environment-induced transcript response in these organisms, but they just have a capacity to adapt to a wide variety of different conditions. And that is, for example, what is shown here. So there's an awful lot of different genes that are important for organisms to survive here. This organism, for example, needs nitrate respiration, it needs heavy metal resistances, it needs to have stress response functions, it can form biofilms. Sometimes we find flagella that are important and so on. Okay, so that tells us, hey, what the cells are doing is not necessarily that they cannot respond to a soil. No, they try to adapt very quickly. And what is interesting, actually, when we looked at the central metabolic pathways in this organism for their aromatic substrate that we give, is that we see that compared to its total metabolic map here in yellow, we see very few changes actually in exponentially growing cells between liquid or these contaminated soils. That tells us that what is important, this organism can adapt its metabolism, and then what it's doing is just the same thing. It doesn't matter if it is in liquid or in the soil, it will adapt itself and then do the same thing. What was also interesting that actually when we inoculate these kind of bacteria, we see a very strong response in the resident community. So these are experiments where we have, again, contaminated soils that we contaminate with toluene, and then we add this bacterium to take care of the toluene. And we look at the metatranscriptomic response of the resident cells that are in there. Not the added Pseudomonas veronii, but the resident cells. And here we're looking at transcripts from this community for oxygenase genes. And oxygenases are typically enzymes that are important for the detoxification of aromatic substances. So that is what this organism is using. Now, we compared here 3 different soils, one where the organism could not survive. So we see very few transcripts appearing. Then actual contaminated soil from a contaminated site where we see lots of transcripts appearing. Okay, and then one from a site that we contaminated ourselves, where this bacteria grew very well, where we also see lots of these transcripts. But now if you look into the transcripts by itself, interestingly, you see that the contaminated site that was aged and contaminated contains a lot of variety of different genes for this type of conversions. Okay, so adding the bacterium here doesn't make much of a difference. But this soil, for example, is incapable of doing this conversion by itself. Still, you see an awful lot of transcripts, but they only come from a step that is very commonly present in many bacteria, which is the conversion of this particular substance that's called catechol, but not the first step. So here, the addition of the Pseudomonas provokes an enormous response because apparently during its degradation of toluene, it produces an intermediate, and then many of the other soil bacteria react to that and start to metabolize it. And we thought that was very cute because it shows that having an inoculant not only can take care of removing the substance, but also providing food and let's say for a shared network later on. So this was very short. I only showed you 3 topics that we are working on. We showed that cultured soil microbiomes can be made. They follow relatively what we call deterministic trajectories that depend on what you put them in. So it's not that a community, that you cannot grow a community that becomes extremely unstable. No, it seems to be following a deterministic trajectory. And that opens up possibilities, I think, also for targeted restoration efforts, where you can think of, okay, degraded soils, we can inoculate them with communities to achieve, better sustained communities. From the observed paired growth of randomly coupled soil bacteria, we see that the types of interactions are mostly favorable on the organism. So favorable that we see that most species that are not able to grow alone can grow when they are partnered with something else. And that's interesting. Maybe the soil is particular that many of those species are auxotrophic and they cannot grow, but maybe this is also something that is just interesting to keep in mind on when we talk about interactions in communities. And finally, we see that inoculants, yes, they can be used to complement specific metabolic functions, at least in soils. That is, I think, hopeful for any of these efforts that work with inoculants. And we also see evidence for a potential cross-feeding network with the rest of the community. And that would also be interesting because that shows that an organism is then embedded into the actual microbiome. So with that, I'm happy to conclude. Thank you for your attention. I'll be happy to answer questions.
Thank you, Jan. If there are any questions, please either raise your hand or unmute yourself or write in the chat. First question from Camille, please.
I'm wondering if you're looking at interactions with plants. I would expect, so for example, if you want to restore a soil, if you use a certain plant that has certain microbe that is useful to restore this, are you looking at plants?
So we ourselves are not a plant lab, but we are working together with plant people. So one of the things that interested us is also how certain bacterial inoculants can actually favor plant growth. So we have been using with that other team, for example, these stabilized natural regrowing microbial communities to see if they can sustain growth, and also if they're adding specific roots. Colonizers, for example, helps in plant growth. That seems to work very well. So much less variation than, for example, they see when they take natural soil. So that I think is also very helpful. Obviously, many people are interested in using plants to stimulate something in the underground. And I think that comes from so this sort of conceptual explanation that I try to give on the soil. So the soil is heterogeneous. There's very little carbon available for most bacteria, because most of the carbon that is in soils is very refractory organic material, that sits there for ages, which is also why a soil is a good sink for carbon, but that is only degraded very slowly. So any effort to try and stimulate specific microbes means you either have to provide a carbon substrate or a condition that would then locally stimulate that bacterial population. And roots are certainly one of the ways that you could say, okay, you can penetrate that heterogeneous environment and try to get to a level where the root can excrete something that allows you to stimulate a pocket of microbes, which is there. That is one of the big problems in this whole soil management. So plants provide local pockets for substrates, and that stimulates largely what this soil bacteria are doing. So that is very well known. To combine this effort can be counterproductive, because when you stimulate with the plant, it provides, let's say, organic acids or so, but that is maybe not necessarily the type of carbon compound that is then unique for the bacterium that you want to introduce. So that can be counter-stimulating.
That's what I was wondering. If it is well known that one type of bacteria is associated to this type of plant, and you know that this bacteria could be useful to restore this type of soil, then you could use this plant.
Yeah, so there's some people that have said, for example, Pseudomonas putida is a root colonizer, so you can use the plants to favor you can maybe even co-inoculate it with seeds of plants and then have like a favorable environment for those Pseudomonas putida. And if these ones also carry a metabolic capacity to degrade a pollutant, then you can bring them into the place with the plant root and then have them stimulating, sort of give them some extra energy. I think that works to some extent, maybe not completely, because you have to compete then with natural occurring pseudomonads, which are very abundant, obviously, in soils. So there's a bit of a trade-off always, I think, there. And it's not really well understood how to engineer this and how to create this thing of having a specific habitat that would then only stimulate the bacterium that you're interested in.
Thank you.
Thank you.
Karina?
Hi. I'm curious to know when you see all these positive interactions, which I find really interesting, and that when you compare how they grow in monocultures versus in the co-cultures, if you start with the same cell density, because some bacteria do take longer to start growing at very low cell density, and if you took this into account.
Yeah, so the thing I think when you work with a community like from the soil is you cannot get monocultures, right? Because if we try to culture isolates from the soil, you only get like 5% of things that are growing compared to all the cells that are there. So if you use this mixture approach, then the disadvantage is you have like a completely random way of encapsulating. That has an advantage and a disadvantage. So the advantage is that in the same mixture, if you try to get 2 cells in a bead, on average, you will have a proportion that has only 1 cell and a proportion that has 2 cells, and then maybe that has a little more. So you have an internal control to your system where you can say, okay, I can look at all the ones that have only 1 cell and study how well they can grow and compare that to the ones that have 2 cells from the beginning and study how well they grow. So the disadvantage of that system that we have currently is we have no clue what is in there. So we cannot determine in individual beads, unfortunately, for the time being, like, okay, oh, this was a particularly good, well-growing combination. What was it? You know, was it bacterium A and B? That for now we cannot do. We are working on that, but that will require some more time. So we can only see these sort of global effects. But interestingly, so if we look at the global diversity of these kind of incubations, we see that we're able to culture phyla that normally we cannot grow on plates. So by having them in beads apparently and partners, then you can see, yes, you can maintain them. So possibly there's a lot of what I said, auxotrophy among bacterial, natural bacteria in the soil that normally would prevent them from growing on plates or whatever that they're doing. And you can capture them back in this kind of community growth or paired growth. Yeah.
Okay, thanks.
Thank you, Jan.
Welcome.
Now
Thanks a lot.
we have to move on.